Noah's really at the forefront of the intersection between the microbiome and immunity, and in a variety of fronts of immune interactions. You're fast. So also, you know, with infectious disease immunity, and hopefully more and more so looking at anti-cancer immunity. So Noah.
Thank you very much. Everyone can hear me? Good. This is going to be quite a big shifting of gears here. So as Marcus said, and thanks for the introduction, thanks for the invitation to share some of our work with you. I was hoping that I'd have some cancer stuff ready for prime time. We have one project that Marcus knows about that we're really excited about. But today I'm actually not going to talk about cancer at all. I'm really going to focus on the microbiome. And I'm going to spend most of the data in the talk talking about the work we've been doing recently, thinking about the effects of small molecules produced by the microbiota on various aspects of host biology. But before I get into that, I thought for this audience, it might be nice to both go through a little bit of background, because you probably don't spend all of your time thinking about host-associated microbial communities, but also try to focus a bit on what I think of as the major challenges in this field, and the way that we're starting to try to approach those challenges, which I hope at least some of the time is somewhat unique as compared to what others are doing in the field. So by now, probably all of you are aware that we're constitutively colonized by trillions of microbes at all barrier surfaces, most notably in the gastrointestinal tract, where we have more than 10 to the 14th bacteria at any given time. And so some people have suggested that we really should consider ourselves as a superorganism that's comprised of a combination of our Homo sapiens cells, as well as these trillions of microbial partners, including both bacteria as well as other microbes like fungi, viruses, phages, and even protozoa and large parasites in the developing world. I'm going to focus today on bacteria. That's where most of the work in this field has happened, and where my lab is focused as well, where it's known really because of revolutions in next generation sequencing, which allow us to very cheaply and easily determine compositions of microbial communities. It's now known that each person in this room contains a unique consortium of at least 500, maybe upwards of 1,000 strains. That assembly of organisms results from this combination of factors that I've kind of listed up here, and people like to throw out these factoids to be impressive in terms of the numerical value, like the fact that there are more bacterial cells in our gut than there are in our Homo sapiens cells in our body, as well as this last fact, which I will dwell on for 1 second, because I've kind of gotten tired of all of these prior facts myself. This last one really still impresses and intimidates me. And that's the fact that there are 150 times more genes in the microbiome in an individual human's microbiome than there are in the human genome. And because each of us have a unique microbial community in our gut, the collective human microbiome likely contains probably 10,000 or more times more genes than we have in our own genome. And if you think about the field, and it sounds like a prosaic goal, but really the goal of this whole field that I'm a part of now, the microbiome field, which continues to grow, is to figure out what all of these genes do, right? What are all of their functions? And if you consider the fact that we have now, I think current estimates say 22 some odd thousand genes in our own genomes, and after decades and decades of intensive research, using every possible method, we maybe understand what, say, 2,000 or something do decently well. The magnitude of this problem really, really becomes intimidating. And so I think one of the solutions to this, I'll try to tell you about. I probably won't have time to go deep into it today, but one of the solutions we're really pushing hard on in my lab is to leverage new developments in high throughput experimentation enabled by next generation sequencing, as well as combinatorial biology, to have our approaches, experimental approaches to this problem, match the scale of the problem itself. Okay, so because it's another thing probably many of you have heard about and read in the news, because it's become cheap and easy to sequence microbial communities and find out who's there. Basically, at this point in time, every disease under the sun, people have taken healthy individuals and sick individuals, sequenced their gut microbiomes, and inevitably they find some difference. And I've listed just a few down here, but these range from things that are somewhat obvious, like IBD, which I'll talk about a bit today, and my lab has focused on, where it's obvious how IBD would potentially impact disease, all the way to things like autism and depression. But the thing that started to frustrate many of us in the field is that for quite a long time, and even really still today, the vast majority of these studies remained observational and therefore remained correlative. And because the microbiome is so diverse and dynamic and also so sensitive to various environmental inputs, there are a huge laundry list of reasons why these correlations could be just red herrings. And what we're really interested in, of course, is identifying changes in microbial communities that aren't just red herrings or even results of disease, but are actually causes of diseases themselves. And so starting about 10 years ago, a group of us around the world that I became part of started to really push hard to try to develop experimental systems to try to transition this field from simple observational correlative studies towards actually being able to potentially prove causal roles for particular microbes or microbial communities or assemblies of microbes in shaping host physiology in health and disease. And the approach that we began to take in close collaboration actually with Andy Goodman here at Yale, who's been a real pioneer in these areas and started on this when he was a postdoc in Andy Goodman's lab, really I think was one of the first people to really start the train moving in this direction, is really a reductionist approach, where what we do is we take advantage of our recent ability to actually capture more and more of these commensal microbes, which were previously thought, many of them were thought to be unculturable. We now know that not to be true. We're able to capture more and more of these microbes in monocultures in what Andy called gut microbiota culture collections, which sounds very fancy, but essentially is just a collection of plates that are in the freezer, each well of which contains one strain isolated from each individual. And then we can use these collections to go in and pick out bugs and test hypotheses. And the other tool we use to test these hypotheses is the tool of so-called germ-free mice. So these are mice that are raised without microbiomes. They're raised completely sterile. We grow them in, breed them in bubbles. All their food and water is sterilized. And so they act kind of like blank slates onto which we can paste these individual microbes or groups of microbes to test hypotheses in a really reduced way. And obviously, there are lots of things that are artificial about this system, but it allows us really to pin down truly microbial contributions to phenotypes and separate them from all of the complications that exist on this other side of the slide. Okay, so we've had actually, I won't really talk much about this today. A lot of this is published. There are some new details we're working on now, but the basic concept is published of this slide. But we've had some success actually in using this approach to identify potentially disease-causing or disease-driving organisms, particularly in the case of IBD, where we've been able to go in, actually generate culture collections from IBD patients, use techniques I won't have time to go into today that leverage the immune response itself to generate hypotheses about which specific microbes in these collections might be driving disease, and then take those microbes, put them into germ-free mice, and find a few very exciting cases where it looks like individual microbes from IBD patients are actually sufficient to either sensitize mice to disease that's induced by, say, a chemical, and even some extreme cases where we find individual microorganisms from people with IBD, where addition of that organism alone to a healthy microbial community from a healthy individual makes that community cause spontaneous development of colitis, even in wild-type mouse models. Suggesting that in some small subset of individuals, I'm not saying this is all IBD, I don't want to overstate the point, but in some small subset of individuals, the fact that they have that bug alone, that they're unlucky enough to have that bug alone, may be the reason that they have IBD. And we are pursuing possibilities, and I guess this is my disclosure, I have a startup company pursuing the idea that elimination or neutralization of those particular microbes could potentially then cure disease in those patients. And this is a real example actually where we took a community of non-cholitogenic microbes and took one microbe from an IBD patient, put it on top, and it caused profound colitis in this case. Okay, so at this point, we kind of felt like we'd started to generate this kind of paradigm for not only transitioning from correlation to causation, but starting to really mature this field to a field that where we could dig down and pin down molecular mechanisms by which microbes were causing disease, and eventually that this would allow us to do rational drug development where we would target either these microbes or the interface between host and microbe to develop actual cures for microbiota-driven diseases. But I have to say, actually, once we started to try to run through this pipeline, we very quickly, or at least I very quickly, became quite frustrated. And there are two reasons for that. One, which is a bit more prosaic, and the other, which, if not profound, is at least to me a bit more problematic. So the first one is, I already told you, for a variety of reasons, most correlations aren't causal. And there are lots of red herrings. And therefore, despite our best, all of our best efforts, when we would run through this kind of culturomics germ-free mouse pipeline, most of the time we'd be wrong. Most of the times the microbes we thought might be doing something would do nothing, right? 9 times out of 10, we'd still be wrong. And that's problematic because while I can draw this simple cartoon to say that we go through this process, in reality, this is incredibly laborious. I didn't maybe highlight this, but most of these microbes are obligate anaerobes. They all have to be grown with specialized media in anaerobic chambers, cost lots of money. Every germ-free mouse that we use, every single one mouse, it probably costs $500. So the fact of the matter is if you go through this 10 times, you can spend millions and millions and millions of dollars. And I can tell you that by spending even, $10, $15 million, you still only come up with a couple microbes that are really interesting. So that's fine for a company, but not so great for my academic lab to spend $20 million and $20 million later find one thing. So that's a little problematic, but still you can spend your way out of that problem. The thing that bothered me more actually is when we tried to start transitioning to mechanism. And here, the problem we ran into was much more troubling to me. And that is that we realized very quickly that even though we, like many others, use ostensibly unbiased technologies to study this problem, things like so-called shotgun metagenomics, where you look at all of the genes in the microbiome, or shotgun metabolomics, where you look at all of the metabolites, that we were really severely limited by prior knowledge. So for example, metabolites, which I'll tell you more about, probably 99% of the small molecules made by our gut microbes are completely unannotated. Most of them are actually even unannotated structurally, but probably 99% are unannotated functionally. And what this leads to is many of us in the field end up actually converging back on what are already well-studied pathways. So even though we go out looking for new things, doing these unbiased technologies, look at all the metabolites, all the metabolites, all the metabolites, in the end, somehow we always end back at the one metabolite that you would have looked at if you chose one metabolite to look at, right? And so to me, this is kind of infuriating, because what's exciting about this field, or the most exciting thing about this field, in my opinion, is we know there's all this dark unmapped space. My lab now focuses really on mapping interactions between hosts and microbes. I would argue that 99% of those interactions, maybe more, are unmapped. But we have these little tiny specks of light, which are the anecdotes we've stumbled across for one reason or another, that show that these microbial communities can have profound effects on our biology. And so if you believe that to be the case, then what could be more frustrating than to continue building up knowledge higher and higher and higher and higher in one tiny corner that we already know a lot about, and not actually accessing any of that darker space? And so we actually decided, in a way, to go back to the drawing board and think about how we could restructure how we even approach these problems. And that's where we really decided to kind of flip everything on its head and try to focus rather than focusing on correlations first, find ways to actually focus on categorizing and characterizing mechanisms first. And the way we do that is by developing new technologies. I won't have any time to go into any of the details today, but new technologies that allow us in ultra high throughput to experimentally interrogate all of the possible ways that microbes can potentially interact with their hosts molecularly. So we have a technology we're developing in collaboration with Aaron Ring's lab, who some of you may know, that allows us to pan individual microbes against all host extracellular and secreted proteins to see all the ways that they could potentially physically bind to the host. Today, I'll talk to you instead not about microbes themselves, but actually about microbial products. And there are a couple, but before I dig into that in the last 5 minutes to really finally show you some data, I'll tell you 2 advantages to this approach. And maybe it also links to Michael's talk as well in a nice way in terms of thinking about patient categorization. One is that when we go from mechanism to causation, we're right most of the time rather than wrong most of the time. If you find a mechanism of interaction, you put that bug in a mouse, it does something to the mouse. It's pumping out millimolar quantities of bioactive chemical. Of course, you put it in a mouse, it does something to that mouse. Satisfying, right? But the more important thing is when you start to look at correlations. And that's because having this knowledge of molecular mechanisms of interaction allows you to ask a totally different question, which is in which subset of the population is this mechanism operable? And that can be as small as one individual. But in that one individual, that mechanism may be the driving force behind their disease phenotype. And you can imagine in many cases, you'd hope, particularly for complex diseases, like inflammatory bowel disease, that it would not be just one individual, but some sort of bin of 2, 3, 4% of individuals that we all, I think, believe IBD, there's probably 20 or 30 core etiologies. And so we're excited about the idea that understanding this mechanism will actually allow us to subset patients into subtypes that share core etiologies based on these molecular mechanisms of host microbe interaction. Okay, so in the last few minutes, I want to run through at a very high level a couple pieces of data that are a result of a functional profiling effort exploring the microbiota metabolite space. So I already said there's a huge number of metabolites produced by the microbiota. The vast majority are unmapped. If you ask people just trying to count and categorize microbes just based on their structure using mass spec, they can't even tell you exactly how many there are. They just give you an answer like there's at least 100,000 features, meaning peaks on a mass spec. But suffice it to say, we know very little still about these chemicals, but we do know from anecdotes that they can potentially impact all aspects of our biology. And indeed, some of these chemicals can actually accumulate in the circulation at levels up to millimolar levels, so exceeding that of almost any medical drug that is currently given. And these chemicals, I should say, result not only from the de novo synthesis of compounds from core building blocks by bacteria, but also from the breakdown of all of the chemicals that we consume, whether they be dietary chemicals or notably medical drugs, which again, Andy Goodman recently has done beautiful work looking at the degradation of medical drugs by gut microbes. Okay, so this is the idea that this is a vast untapped resource, but how do you identify the biologically relevant compounds when they're hidden in this vast sea of irrelevant chemicals? I already told you the solution. We use functional profiling. In this case, what we decided to do was focus on the largest family of potential sensors of small molecules encoded in the human genome, which are the 7 transmembrane receptors known as G protein coupled receptors, which probably many of you know about already for these reasons I've listed on the left here. Suffice it to say that this was kind of the obvious place to start because it's already known that there are gpcrs that sense microbiota metabolites, and also because this is such an expanded family that regulates so many aspects of our biology, from vision to taste to nutrient sensation to immune cell recruitment. And so what we decided to do a few years ago, and when I say we, I really mean an incredibly talented postdoc in my lab and brave postdoc in my lab, Highway Chen, is take advantage of our ability to culture many microbes that I already told you about and combine this with a new technology that had just been published at the time that allowed us to simultaneously look at the activities of all conventional GPCRs, more than 300 conventional GPCRs, in a collection of 496 well plates. And so I won't go through any of the details of this at all, other than to say basically what we did is take supernatants from bacteria, so the mixtures of chemicals that they're making, and ran them through this assay to look at all potential activation of, or the potential activation of any conventional GPCR. What comes out is this horrible kind of, you know, the data that comes out at the end of a lot of these kinds of experiments is you get this horrible heat map that basically means nothing. There's lots of interesting stuff there, but it's totally uninterpretable, at least by eye to me. So we have to figure out ways to visualize this. The best way we've figured out so far is looking at these kinds of dendrograms. You don't have to read any of the tips here. Basically what this shows, the only take-home message, is that the microbiota is a remarkably rich source of bioactive metabolites, including many, many GPCR ligands, including actually, so the color at the tip means the fold activation by any microbe. The size of the circle actually means, represents the number of microbes that activated that receptor. And you can see that there are hits across basically every family of receptor, ranging from the aminergic receptors like the dopamine receptors, all the way to hitting the orphan receptors. So I'm going to tell you just just 2 or 3 slides, a couple of interesting anecdotes that came out of these studies. So the first one is actually that we found a microbe that's making this compound called phenethylamine. And phenethylamine is a trace amine. I didn't actually know anything about it at the time. That has potent mood enhancing effects. But the key thing about phenethylamine is at the bottom there. Unlike dopamine and tyramine, it can actually readily cross the blood-brain barrier, which raised the possibility that you could actually have a mood-enhancing compound being made in the gut, making it all the way to the brain. And indeed, we were able to show, skipping over a bunch of details, that this is published, so if you want to go look it up, I'll give you the reference at the end. Skipping over a bunch of details, we were able to show that when we colonized mice with this microbe making phenethylamine, and we gave them an antidepressant that inhibits monoamine oxidases, which normally degrade biogenic amines. But it's an antidepressant that's still in use. It was the first FDA-approved antidepressant. Many of you may know that. Now fallen out of favor and mostly replaced by SSRIs, but still used. What we could find was that only in microbes colonized with, only in mice colonized with this microbe and treated with MAOIs could you see profound accumulation of PEA in the brain. And these mice actually died of phenethylamine overdose and were having seizures when they died. Now, I should say we don't think this is happening in humans. This is totally artificial. Mice are colonized with this one microbe. It's filling up their whole gut. Normally, this microbe would be less than 1% of the overall community of an individual. But it does raise this interesting possibility, which we're interested in exploring further. I have no real in-person evidence for this yet, but I'm interested in looking at it, that actually presence or absence of this kind of microbe might explain inter-individual diversity in responses to medical drugs like antidepressants. You could imagine a very simple scenario. You have this bug in your gut. It's making this happy-making chemical, but that's being degraded by your own enzymatic machinery all the time. If you de-repress that by giving MAOIs, all of a sudden you're happy as a clam. If you don't have this bug in your gut, you can take as much MAOIs as you want, and all you get are side effects. And in fact, many of you may know that antidepressants often work that way. Some people, boom, it's great. Other people, nothing at all other than all of the unhappy side effects like GI toxicities. Okay, we also find, I'll just mention, many, many orphan GPCRs that are activated, so this is a really rich area. We were able to de-orphanize, or we've done more by now, but one that we stumbled across was actually, we found a new essential amino acid sensor. And we stumbled across it because we discovered this very weird microbe. We've never seen any other microbe like it that's pumping out the essential amino acid L-phenylalanine. Okay, so this is what I told you about so far. We also stumbled across, interestingly, and I won't have time to go into details, something many of us in this field are really interested in these days, which are metabolic exchanges, or sometimes referred to as cross-feeding between bacteria, where you have one bacterium that's making a precursor that's processed by another bacterium into a bioactive compound. In this case, it's this pathway I just told you about, where we found a B theta strain making L-phenylalanine, and a Morganella morgani strain that takes this L-phenylalanine and makes it into phenethylamine. And we can demonstrate that that exchange occurs both in vitro and in vivo. So hopefully these are the conclusions. Hopefully I've convinced you that, gut microbes are a really rich source of potential bioactive compounds with lots of potentially interesting effects. including agonists for orphan receptors. And I should mention, I'll mention at the end as well, that all of this work was a really close collaboration with a chemist here at Yale, Jason Crawford, who's at West Campus and really was an integral part of all of the chemistry that I didn't have time to show you behind this. The second point, hopefully, I've gotten across is that this, what I call functional profiling approach, and these kinds of new experimental approaches that we work on in the lab are useful ways to start to uncover causal and mechanistic roles for gut microbes and their products in shaping host biology, including human biology. I should emphasize again that all of the microbes we've made a choice in the lab to focus all of our work on microbes isolated from humans. And we basically don't work on mice at all, other than using them as receptacles for these microbes that we get from the human population. The final thing is that I think we've really only scratched the surface of this field. And I've put up this totally cliche picture that's probably terrible to put up at all of the iceberg. But I think if you're to represent this properly, really it's only the tip of the iceberg that we've really looked at so far. And there's a lot more to do. And one of the things we've been working on for the past couple of years in my lab is developing ever more high throughput, ultra high throughput technologies to interrogate more and more of this space, and hopefully eventually enable us to mine if not the full depths of the microbiota metabolome, then at least get below the surface of the iceberg. So with that, I'll finish and just say thanks for the opportunity to talk to you guys. Again, I want to emphasize that this really was all driven by an incredibly brave postdoctoral fellow, Hiwei Chen, who's pictured here on the left, and really critical contributions, particularly from a number of individuals, but particularly from Jason Crawford's lab, and Phuket, who's a really great graduate student and really talented graduate student in Jason's lab, who probably did 2,000 LCMS samples that lie behind all of this data that never get shown, but of course were critical for all of this to be believable. So yeah, with that, I'd be happy to take questions.
One step, though, which I imagine must be a pretty major bottleneck. How do you identify and purify the agonist that's sitting in gpcrs?
So that is indeed a major bottleneck. And the way we do it is also a black box to me, because this is all really done by Jason. Jason, some of you may know, Jason is an absolutely brilliant natural products discovery chemist. And so we go through, we do fractionations, fractionation-based natural product discovery, NMR, all this kind of stuff. And your point is spot on, because this is actually where we most often get caught up. And really so far, one of my big disappointments, I'm sure we'll get past this, but is that so far, most of the compounds we've actually uncovered are quite high abundance in the supernatants and quite low affinity. And the reason we find them is because they are low affinity. And so they're low affinity, so they have to be high abundance in their, high concentration, and that makes it easy for Jason to find them quickly. For the high affinity compounds that are hidden down there in the depths of that sample, it's a much bigger problem. So we're exploring various methods to potentially supplement some of these studies with approaches where we actually start with chemicals that we know microbes are making, rather than starting with these super complex mixtures. Then you're restricting yourself, which is disappointing, but you may save yourself 2, 3, 4 years of natural products discovery efforts that end with nothing. Yeah, or we can give Jason more money, yeah.
One quick question. So for the natural products that are made by the bacteria, I imagine that some overlap with things that might be produced in humans, for instance, phenylalanine and so forth. So how, are you focusing on ones that are more unique to the bacteria that don't happen in humans, or how do you make those decisions?
Yeah, so this is a really excellent point and excellent question, and actually gives me an opportunity to say just a few words about something that I cut out, but that I usually talk about in talks, which is how my thinking about this conversation between host and microbe and how that evolved has actually changed because of this work. So most of the chemicals we actually find that are bioactive are chemicals that we also make or that are common in biology in general. And what this has made me realize is, or maybe it's obvious to other people, but it wasn't obvious to me, is it's really underscored how much common chemical currency there is in biology. And that common chemical currency probably results from how evolution works, which is that once we've invented something important, or something useful, sorry, whether it be a gene or a chemical, it gets used and reused for different purposes over evolutionary time by different organisms. And in fact, you'll sometimes read stories about bacteria making neurotransmitters, right? As if bacteria have decided to make neurotransmitters. Turns out if you reconstruct the evolutionary history of neurotransmitters, all of them were made first by bacteria, evolved first by bacteria. The original evolution of dopamine is that bacteria figured out how to make dopamine, entire biosynthetic gene cluster. At some point, the model was acquired by eukaryotes by horizontal gene transfer. We took the ability to make that useful chemical, and we built our whole nervous system around it, right? Because it's useful. But what that means is that basically, I haven't figured out the best way of saying it, but basically what it means is that we use the same words to actually speak different languages. And that makes, to me, in some ways, even though it changes the way you think about the evolution of the system, it actually makes probabilities of chemical collisions between host and microbe even more probable and even more common. And so I think there's some sort of language but non-shared language, and that a lot of this is then coincidental things running into each other. There was no plan for them to run into each other, and that can either lead to pathologies, or sometimes it may lead to good things, and then that itself can get selected on by or get selected on by natural selection and become fixed.